Assalamu alaikum ji. Today we will discuss use of an EPG. So let me open the use of an EPG. Okay, so before we discuss this use of an EPG, we should understand what does an EPG mean. You can see in this picture or in this image, you have this an eyepiece which is fitted into a microscope, and you view through this an eyepiece. Here, inside it, you can see this is a glass which has a graticule printed onto it that has been shown here in this diagram. So this is a graduated graticule. Okay, this is an unknown length of each division, so this is an unknown scale. Okay, so it doesn't have any fixed scale. This is used to compare the proportions in size of the different images, and is also used to measure the actual size or actual diameter of an object. So let's see, when we view it under a light microscope through an eyepiece. So this is how we will see this an eyepiece graticule in a field of view. This is field of view. Okay, and this has 10 larger divisions, and each division is further divided into 10 smaller divisions. So as a whole, it is divided into 100 divisions. But what is the length of one division is not known, so it is an unknown scale. To calibrate an EPG, we use another scale which is called stage micrometer. You can see this is a stage micrometer slide. It has a scale printed on or engraved on it, or maybe embossed on it. So this scale has, stage micrometer has a known scale, and this known scale is generally printed on a glass slide or maybe given with a pointer. In this case, this is 0.01 millimeter printed on it. So this means one division of SM is equal to this 0.01 mm. It may be 0.1 mm, or it may be 0.01, or it may be 0.02 mm, depending upon the different scales or stage micrometers we are provided. Generally, in our syllabus we have this particular stage micrometer which has one division measuring 0.01 millimeter. So let's see, this stage micrometer is placed on the stage of a light microscope, and when we view through an eyepiece, we will see both an eyepiece graticule, this is an EPG, and we will see an SM as well. So this is an SM. Both will be visible in a field of view. They may not be parallel initially, but by rotating an eyepiece and by moving the stage right and left or back and forth, you can bring both of these both of these graticule and scale parallel to each other. Once these two are brought parallel to each other, we will have to calibrate an eyepiece graticule. So this is step number one, which is called calibration of an EPG. In this case, we have to find what is the length of one eyepiece graticule division. So this is, for example, one eyepiece graticule. This is one EPG. We do not know what is the length of this one EPG, but however, we know that the length of this one division of an SM, which is 0.01 mm. Okay, so to find how many divisions, of EPG coincide with or overlap with how many divisions of SM, we have to find the coinciding lines. You can see this line is coinciding with this, and this line is not coinciding. Okay, so you see this line is not coinciding. So we can find some other line which is coinciding. So this line is coinciding with this, and this line is coinciding with this. So we find two coinciding lines, and we count the number of SM divisions from here, which is 1, 2, 3, 4, and 5. So these are 5 divisions of SM. And from here, if you look at this, we will have to count these, divisions from here. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, and 19. So these are 19 divisions of EPG. So the point is 19 division of EPG are equal to 5 divisions of an SM. So let's see how we make calculations for calibrating the EPG. If 19 divisions of EPG are equal to 5 divisions of SM, we already know one division of SM is equal to 0.01 mm. So 5 divisions of SM will be equal to 0.01 multiplied by 5. So this gives us 0.05 mm. So putting this value of 5 divisions of SM here in this equation, we will have 19 divisions of EPG are equal to 0.05 mm, right? So now we have to calculate how many divisions okay. So we have to calculate how many mm would be one division of EPG equal to. So one division of EPG is equal to 0.05 divided by 19, and this would give us 0.0026. You can write up to 3 decimal point or 4 decimal points, so that doesn't matter because this is an intermediate calculation. We have to use it later, so there is no need to round it up or round it off. We can use this value as such. So one division of EPG is equal to 0.0026 millimeter, or we can convert it into micrometer by multiplying it with 1000. So this will give us 0.0026 multiplied by 1000. So this gives us 2.6 micrometer. So this is our calibration. Now, once you have calibrated one division of, we know what is the length of one division of an EPG. we can measure the length of, or we can calculate the actual size of or actual length of a cell. We can take an example. Now, in the second step, this is step number 1, and this is step number 2. In second step, we remove this SM and replace it with a specimen slide. So this is a specimen slide. Obviously, when the stage micrometer has been replaced with the specimen slide, now the specimen will be visible instead of an SM, and EPG will be visible there too in the second step. So this is an EPG visible here, and we have this specimen visible here. So we can use it to count the number of divisions of EPG overlapping from one edge of a cell to the other edge of a cell. Maybe we can take an example of this cell. Okay, so maybe this is an example. So it has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, and 18. So this is 18 divisions length of this cell. And we know if it is 18 divisions of EPG, the length of one cell, we know the length of one division of EPG now from this step where we have calibrated one division of EPG. So when we know one length of one division of EPG by multiplying it with the total number of divisions of EPG overlapping from one edge of this cell to the other edge of a cell, we will get the total length of the actual length of this cell. So 18 multiplied by 0. Or multiplied by 2.6 micrometer would give us 47.36 micrometer. So the length of this cell is 47.36. Now you can round it up or round it off. Maybe you can write 47.4, or maybe you can write 47. So this is 47 micrometer length of a cell. You can calculate the mean length of a cell. So that's how an EPG is used to calculate the actual length of a cell. Let's see a past paper example question. Just give me a minute. Let's see. Okay. So let's see this question. This diagram shows a stage micrometer scale viewed through an eyepiece containing a graticule. The small divisions of the stage micrometer scale are 0.1 millimeter. So in this case, the one division of stage micrometer scale is given here, and it is 0.1 millimeter. Okay. So step number 1 is calibration of this EPG. So this one is an SM, and this is an EPG. So one division of this SM is equal to 40 divisions of EPG. So we can write an equation here. 40 divisions of EPG are equal to, you see, this is from here to here. it is 0.1 millimeter. So 40 divisions are equal to 0.1 millimeter. And one division of EPG would be equal to 0.1 divided by 40. Okay. And that gives us 0.0025 millimeter. We can convert it into micrometer by multiplying it with 1000, and 0.0025 multiplied by 1000. This gives us 2.5 micrometer. So one division, this is the calibration here. Okay, so one division is calibrated. So one division of EPG is equal to 2.5 micrometer. So we will have to use these calculations in the next step. This is step number 1, calibration of EPG. In the second step, which is given here, we remove this stage micrometer. This stage micrometer has been replaced with this specimen slide, and this single plant cell is visible in this image. Here you can see this is an EPG, and the question is the stage micrometer scale is replaced by the slide of a plant cell, and what is the length of the nucleus in the plant cell. So we have to find or calculate the length of a nucleus. To find that, we will have to identify nucleus. So see, this is a larger densely stained body in this plant cell. The rest are very smaller. So the smaller ones are chloroplast, and this larger one is the nucleus. So look at this nucleus. Measure the number of divisions of EPG overlapping from one edge to another edge. So from here to here, this makes around 9 divisions of EPG. So from one edge of a nucleus to the other edge of a nucleus, there are 9 divisions of EPG overlapping. We already have calculated the length of one division of EPG. So the diameter of this nucleus will be, or the length of this nucleus will be length of one division, which is 2.5 micrometer, multiplied with the total number of divisions overlapping from one edge to another edge, multiplied by 9. And this would give us, okay, this will give us 22.5 micrometer length of a cell. So we will have to find the closest answer. Why we need to find, why this answer is not found in these 4 options? The reason being, when we are finding the overlapping divisions from one edge to the other edge, we need to place a ruler here so that we can exactly find the margins, and we can count the number of divisions overlapping from here to here. So there may be an error of +1 and -1 divisions when we are counting the number of divisions overlapping from one edge of an object to another edge of an object. So considering that this answer 22.5 is closer to 25 micrometers, so C will be the correct choice in this case. Okay, so that's how you will have to attempt such questions in your exam. You can have more questions on this topic on the use of an eyepiece graticule. So here you have the eyepiece lens of a microscope can be fitted with an eyepiece graticule. Which of these statements about eyepiece graticule are correct? They measure the actual length of the cell in micrometer. No, they don't measure the actual length of cells in micrometer. Why? Because an EPG doesn't have known scale. EPG doesn't have known scale. So they are not used to measure the actual size in micrometer. Instead, they can be used in calculations of the actual size of an object. by first calibrating them and then by multiplying it with, the number of divisions overlapping with an object from one edge to another edge. So this is not a correct statement. The second one is they help biologists to draw cells with correct proportions. Yes, you can use an EPG to draw the different cells or different objects with the correct proportions. Okay. For example, in this case, let's see. In this case, you see, if we have to find the proportion of the length of a cell with the length of a nucleus, we can count the number of divisions overlapping from one edge to another edge of a cell. We can count that from here. And similarly, we can count the number of divisions overlapping from one edge of another edge, one edge of the nucleus to the other edge of it. And we can count that there are two divisions overlapping with it for a nucleus. And there are many divisions, we can count them, whatever is the exact number you can count from here, or we counted it in a previous step from this cell. Okay, starting from here to here, which was 18 divisions. So the ratio will be length of cell will be 18 divisions and the length of nucleus, this is length of cell, is 18 divisions, or length of nucleus is 2 divisions. So this is how we can have the proportion of these two structures, or any other two structures can be compared by counting or by measuring the number of divisions overlapping from one edge to another edge, and this can be used to compare the proportions. So this statement is true. So they help biologists to draw cells with correct proportions. We can find the proportion. For finding the proportions or comparing the proportions, we don't need to make measurements or make calculations. Instead, we will just count the number of divisions overlapping from one edge to another edge of one object, and similarly for another object. So they change in size when the objective lens is changed from X10 to X40. As an EPG is fitted into an eyepiece reticule, so changing the power of an objective lens will not change, the size of an eyepiece reticule. Size of an eyepiece reticule will only change if we change the power of an eyepiece reticule, eyepiece lens. If we change the power of an objective lens, then the size of an eyepiece reticule will not change. So this is also not a correct statement. So we have only two correct statements. So this D is a correct choice. Okay. So that's how you can have questions on use of an EPG or use of an eyepiece reticule.